Okay, is it available? So maybe we can get started. Hi everyone, thanks for joining our ISBS webinar this month. My name is Henzhou. I'm from Merck. It is my pleasure to be your host for today's seminar entitled "A Quantitative Approach to Maximize the Return on Investment in Oncology Drug Development," presented by Dr. Cong Chen. Cong is the scientific AVP in early and mid-stage oncology statistics at Merck, providing fit-for-purpose decision-making strategies and novel statistical approaches for oncology clinical development programs, and supporting oncology external collaborations, competitive intelligence, and high-profile due diligence projects. Prior to taking the role, he led the statistical support for the development of Keytruda and played a key role in accelerating its regulatory approvals. Cong is an elected fellow of ASA and associate editor of statistics in biopharmaceutical research, a member of Cancer Clinical Research editorial board, and sorry, sorry, there's some glitches, and leader of the DIA Innovative Design Working Group. He has published over 100 papers and 10 book chapters on innovative design and analysis methods of clinical trials. He has also given multiple short courses on this subject at statistical conferences and is a frequent speaker and panelist at major clinical conferences, focusing on statistical design strategies and regulatory policy for oncology drug development. Before we get started, I want to remind you that our webinar is recorded. And as a matter of logistics, during the presentation, I will mute all other audio lines until the end of the presentation when you will be able to unmute your line to ask questions. So without further delay, let's welcome Kang to start his presentation.
Okay, thanks, Han, for the invitation and for the nice introduction. By the way, Han, do you hear me okay?
Yes, pretty good.
Okay, so I'll talk about basically decision analysis in oncology drug development, and this joint work with Linda Sun, my colleague at Merck. So this is just a quick introduction to the background why I'm giving this presentation. As we all know that the immune therapy revolution in the past decade has totally changed the landscape of drug development. In terms of clinical strategies, the highly successful immune checkpoint inhibitors has encouraged extremely aggressive approach to developing not only these agents, but similar immunotherapies. However, the success of new drugs that are rare and infrequent. And as a statistician, we should, I don't need to remind you, but it's just a matter of fact that with the improvement of standard of care, thanks to those immune therapies and other novel drugs, the most of the novel therapies on the clinical development will have a high bar to cross, and they are unlikely to have the same success as those immune checkpoint inhibitors. Therefore, a more balanced, less aggressive approach is probably required. So in my presentation today, I will introduce benefit cost ratio analysis for this purpose. It attempts to maximize returns on social economic investment. The method was originally developed in a similar environment in the pre-IO revolution area, which was about 17 years ago. So my purpose here is to reintroduce this to you to provide provide you with a fresh perspective that can potentially help you when you have to make critical decisions along with your clinical developers. And this is kind of a norm in oncology drug development. In the last decades and decades. Although the IO revolution has changed it substantially, but the norm probably is going to persist moving forward. What I'm trying to call attention is the fact that the cost of these trials from phase 1 to phase 2 to phase 3 just goes up substantially. And some of you who are not in the oncology space probably are not aware of that one single patient probably is going to cost a developer 100,000 to 200,000 patients. So you multiply this by sample size of phase 3, which is in the neighborhood of 1,000, you immediately get an astronomical number. And as a comparison, a phase 2 study may be in the magnitude of 100 patients. And for phase 1, the signal detection is probably even less, but just that the risk in terms of the cost goes substantially up. It wasn't a big deal during the IO days because the success rate was pretty fast. A lot of developers immediately head to phase 3 after phase 1. But now we are in a very different scenario. So before I present any quantitative decision analysis, I have to disclaimer, put disclaimer up front, because as we know, any decision is very complicated. It's hardly quantifiable. And to that, roughly speaking, there will be oncology programs that warrant a more aggressive development plan. And while there are others that may warrant a more conservative one. However, as a statistician, I think you probably will agree with me, majority of the projects will be suitable for baseline decision thanks to the bell curve. So my focus is on those baseline decisions in the middle. And to that, I will apply the so-called benefit-cost ratio analysis. And analysis, I have to credit Bob Beckman, my longtime collaborator. He was once executive director at Merck. And almost 18 years ago, he came up, it's 18. He came to me with a question in the email. The title is a very simple idea. Basically, if I break down that into statistics, it goes as follows. Assuming that you have a phase 2 study on the planning, and typically a phase 2 study may have type error rate of 5% and power of 80%. Of course, in practice, we sometimes also see type error as high as 15%, or for this matter, there are more conservative developers that even use 2.5% for phase 2 study. Of course, it's based on an early endpoint, not the clinical endpoint. And let's assume that's going to cost C2. And once you have a go decision, the subsequent phase 3 study will cost you C3. Usually, the sample size of phase 3 is kind of predetermined in some sense. It's not going to substantially differ from what you see from phase 2. But of course, it can. Just for simplicity, let's put it C3 out there. So with this setup, very simple. What's the optimal alpha of this study? Assuming that a go decision is going to be made if the p-value is less than alpha, and we're sticking to this decision criteria. So this is very classical statistical problem. And if you look at the literature, there are not any good quantitative arguments. Of course, you don't have to be quantitative, but just that most of the arguments, if any, are just very qualitative. That's why the range of alpha is huge. So to address this question, one thing we need to assume is the probability of drug being active with a target effect H1. I assume to be P, which is unknown. I don't even need to know, as you will tell later. You will see later. And as a consequence, I will just also assume that the probability of drug being in active is just simply 1 minus p. Remember, given the sample size constraint, whenever you change alpha, the beta will also be changed, but they satisfy this equation. Of course, Z is just the normal quantile. So the efficiency of this phase 2-3 program is as follows. On the numerator is P, the probability of drug being active, multiplied by the actual power, 1 minus beta. And then in the denominator is C2, which is the cost that you will have to spend to gain this gain some information about the drug activity. And then the plus C3 multiplied by the probability of going to phase 3. And the probability going to phase 3 can be broken down into two pieces. The probability of right decision, which is just P times power, plus probability of wrong decision, which is just 1 minus P times alpha. So it's very simple and easy to understand. And in case you're wondering what the heck is this, you can try to inverse the order of denominator and numerator. You realize that basically this is a measure of expected cost per active indication identified. Apparently, the lower the better. And for this matter, you try to optimize the this ratio, which means that you're trying to optimize the opportunity of getting an active indication per cost of the trial. And of course, if we don't have the actual cost available, you can just use sample size as kind of surrogate. So mathematically speaking, maximizing of this quantity under the constraint on alpha and beta gives the optimal alpha as well as beta. Okay, so what does this exercise looks like? If you look at that equation, it doesn't depend on the actual C2 or C3. It depends on the C2 C3 ratio. So if you look at the C2 to 3 ratio, that's basically what you get. What first all you observe is that consistent with your intuition, when P increases from like 0.1 to 0.3, the optimal alpha also increase, meaning that if you have stronger confidence about drug activity based on prior data, your bar is lower. As you know, a greater alpha means lower bar. But the increase is very minimal when P is in this reasonable range. A typical oncology drug probably only has 10 to 30% of chance to be really active, as we know. And this optimal alpha also increase with the ratio, apparently, and it increase faster. So if we look at the numbers, if you want to find the corresponding alpha, corresponding C2 to C3 ratio at 5% alpha, it's about 0.4. What this means is that if the phase 3 cost is 2.5 times that of C2, the starting point of 5% and 20% is actually optimal choice from this point, benefit cost ratio point of view. And as for a stringent alpha of 2.5%, it corresponds to C2 to C3 ratio of 0.2, meaning that if phase 3 is 5 times expensive as phase 2, you probably should use a more stringent alpha at 2.5. And this on the right panel is just the corresponding power, which is simple consequence. And one thing you may notice is that even when C3 is equal to C2, which is apparently impossible because by definition, phase 3 cost is going to be more than phase 2. But even at this extreme situation, the type 1 error rate should be 10%. So in other words, if somebody, for example, your clinical director is asking you to give to set the bar to be as low as 10% or 15%, you at least should know that from benefit cost ratio point of view, it's a little ridiculous. And in oncology, a lot of developers just directly go to phase 3 based on phase 1 single arm data. That's kind of a common practice in the last decade. This analysis shows that if your phase 2, if your signal detection is based on very small data, and given that the actual type 1 error rate from single arm study is hardly quantifiable, you do need to make sure that the signal is indeed eye-popping before you move to expensive phase 3. Otherwise, it's just not cost effective. Now, with this simple kind of illustration, let's now just try to apply to more examples. The first example is Relativity 47, which is a study sponsored by Bristol Myers. It's phase 3 of RELA plus Nivo versus Nivo in first-line lung cancer, first-line melanoma cancer, and RELA is the LAG-3 monoclonal antibody. The PFS is the primary endpoint. The study has 85% power for detecting hazard ratio of 0.73. The prior data before they initiated this study is very limited, and so they took a more or less cautious approach of phase 2, adaptive phase 2, 3. In the phase 2 portion, they enrolled 400 patients, and they paused for 6 months, and then they check on PFS, and if the PFS has a ratio of less than 0.8, they continue. Otherwise, they will probably stop at the end of phase 2. Of note that the final analysis, the final hazard ratio after the finished phase 3 was 0.78, which was awfully close to the interim futility bar, which they were able to pass, actually. The original plan for this study was actually adaptive phase 2, 3 with go-no-go decision based on ORR of 200 patients. So they changed that to 400 because they were not sure about the benefit of ORR, how it translates into PFS benefit. Anyway, this study actually is quite successful from a high-level point of view because it gives the positive outcome at the end, leads to the approval of RELA 2 years ago, and RELA is kind of the first and arguably the only new immunotherapy that was approved post the PD-1, PD-L1 revolution. So what does if we apply the benefit cost ratio analysis, what should futility bar looks like? So in this case, unlike in the previous example, the actual power, which is the benefit of this study, also depends on the futility bar. Apparently, the higher the bar, the lower the actual power. But nevertheless, the mathematics is the same. And by the time 400 out of 700 patients are enrolled, my kind of guesstimate is that 70% of the total cost probably is spent because due to the trial setup, etc. Despite the fact that the proportion of patients is 60%, probably more is spent. And my guess estimate is that the PFS information is probably somewhere between 40 to 50%. Those numbers are not important. They are just for illustration. And again, if you go with the benefit cost ratio analysis on the assumption about the true activity that ranges from 10% to 50%, very wide range, your futility bar should be set around 0.91 or 0.9, that kind of range, and that represents kind of 1/3 of the treatment over the target effect. The target effect is about 27% hazard reduction, and basically this says that you probably need to have observed 9% hazard reduction before you move forward. But of course, the company took a more aggressive approach. They wanted to observe a 20% hazard reduction. I'm sure there is a good reason for that, but just that if you base upon this analysis, the bar seems to be a little more aggressive. Granted, the original design that would make this decision after 200 patients would favor a higher, relatively speaking, of course, a higher go-no-go bar based on ORR because you have now more cost saving down the road. Cost is going to be a key driver. And as we already see in the first example, I don't want to repeat this here. And for this very study, I would argue that a 2-in-1 design could have been considered to mitigate the risk of potential forced no-go decision. For those who are not familiar with this term, 2-in-1 design is basically kind of a new realization in the adaptive design literature. Basically, it shows that both phase 2 after no-go decision and phase 3 after go decision can be tested at 2.5% level without inflating the overall type 1 error rate in general. And speaking of 2-in-1 design, because I personally believe this has more applications in oncology, I just want to talk about in general which kind of go-no-go bar over there should be set. And just kind of a high level point of view, the 2-in-1 design allows a sponsor to based upon more solid randomized controlled data to make a final go-no-go decision to phase 3, as opposed to the kind of a prevailing approach of just using very limited single arm data to make a binary decision upfront of about whether to go to phase 3 or not. So from a higher level point of view, the 2-in-1 strategy is advancement, and especially given that a lot of programs should probably have done phase 2 in the first place, and a no-go decision during a 2-in-1 design, it doesn't necessarily mean it's a failed study, because statistically you can still declare success, and a sponsor shouldn't be ashamed of having positive phase 2 by any means. So in other words, the adaptive phase 2-in-1 design allows you to use the more solid data to guide you which path to take, whether it's phase 2 or phase 3. Again, that's in contrast to the kind of prevailing approach that's based upon very shaky phase 1 data to decide whether to immediately go to phase 3 or not. So we did a very extensive investigation, and not surprisingly, that it shows similar robustness of the optimal bars to the P, which is unknown. And since in this case, both phase 2 and phase 3 has their value, of course, a positive phase 2 will have much less value than phase 3. Nevertheless, this relative value is also incorporated into the benefit term. So now we are adding one more term to that benefit component in the benefit cost ratio analysis. And when the relative cost is within a relatively representative range, I assume to be like the overall trial cost is 2 to 4 times that of phase 2 portion, or the sample size wise it can be more, but that the fact that you are making this adaptive design means that you have to put a lot of pre-investment. And the outcome is not that surprising at all. Just like in the previous example, the optimal bar basically is empirical bar for go-no-go decision is 50 to 60% of target effect. So if you are targeting 20% response rate for phase 2, then you should probably have observed 10 to 12% of difference before you make such a decision. And the bar basically shouldn't be set too high. So if you are targeting 20%, you certainly don't need to observe 20%. But on the other hand, you don't want to just go with after a positive trend. Now let's take another step by applying to a slightly more complicated example. This is a real study, operationally adaptive phase 2/3 study. I think this sort of design has legs in the current environment of when FDA is emphasizing a lot on Project Optimus. So in this very study, we test the new drug versus doxorubicin in ovarian cancer. The primary endpoint is overall survival. In the first phase 2 portion, we have low dose, high dose, and control. And the phase 2 purpose is for both dose selection, primarily based on safety, and also for a solid proof of concept based on PFS. The drug was expected to have better safety profile than doxorubicin, so we think that the non-inferiority outcome for OS coupled with superiority in safety may still be sufficient for approval, although our primary objective is to demonstrate superiority of OS. So in this very study, the phase 2 portion has 210 patients and with one-to-one randomization, and this phase 2 officially would complete after all patients have had a 4 months follow-up. And if the data is positive and we can successfully make a dose selection, the phase 3 will be initiated with a total of 760 patients. However, we build an upside right after all the patients, and to be more precise, it's 1 month before all the patients have enrolled. We take a look at analysis data. By that time, there will be approximately 140 patients, 150 patients, or well, I got a number. Let me see. It's probably 140 patients with 2 to 3 months of follow-up or something. But if bottom line is that if the data looks very, very promising, we will have a seamless transition to phase 3 because data analysis, etc., can take a month or easily take a month. So that's why we want to do the analysis. The data cutoff actually is 1 month before the patients are enrolled. And our operation expert told us that despite the fact there is just a 4 months gap here, actually, if we can successfully transition seamlessly to phase 3, we may be able to save 9 months down the road because if we pause for 4 months, then there will be loss of enrollment momentum, etc. It can take some time to catch up. Now, in this very study, let me see. The end of phase 2 go-no-go bar is going to be based on the benefit cost ratio analysis. I will show you. That's our focus, of course. And to make this seamless transition, we require that the interim data to give us a 80% confidence of meeting the end of phase 2 go-no-go bar. So the interim data numerically needs to be even better than the end of phase 2 bar, so that we can have 80% of confidence to beat that bar. That's the criteria we pose for seamless transition. So this is set up for PCR analysis. Now we're basically using a surrogate endpoint to mainly derive make such a decision for phase 3 study. Naturally, some of the clinical folks raise the question, well, what about OS data? You know, we can, despite short follow-up, we given the sample size and the relatively long accrual period, we may still have appreciable number of patients who have died, unfortunately. And what about we incorporate this data into the go-no-go bar as well? So to that, we looked literature and we come up with assumption that the grade, which is that hazard ratio for hazard reduction for overall survival, is going to be approximately half of that of for PFS, but plus minus, of course, in this case, plus minus 0.2. So in other words, a 45% hazard reduction in PFS may translate into a hazard reduction OS has a reduction about 20% for survival, plus minus, you know, again, some random variable, some variance. And the benefit component in BCR is now further adjusted by the relative commercial value and regulatory probability between superiority outcome and non-inferiority outcome. So how to get this kind of information? Well, first of all, in terms of prior belief of drug activity, I asked the team, well, since we don't know anything about the drug at that stage of the game, because, you know, there are just some very limited phase 1 data, is it fair to assume that the drug has equal probability to be superior, equivalent, or inferior to the control? Everybody feels like it's a fair assumption because really we don't know. And later on, actually, of course, we did some sensitivity analysis. And as you might expect, this assumption, as long as it's not extreme, has little impact on the go-no-go decision bar. That's kind of, again, I have to emphasize why this analysis does make practical sense, not just theoretical sense. And then relative commercial value, so as well as relative regulatory probability. I conducted a poll. You know, I asked the team members, tell me what's the relative ratio. You know, I provide some numbers like 1 to 10. You pick one and just send to me. And if you are high up on the org chart, you think that your opinion is versus 2, also let me know because I can adjust the outcome accordingly by giving you a weight of 2. You know, and of course nobody dares to claim that they know any better than others. And once I also told them upfront, I will take your geometric mean, and that's how I arrive at 5 and 2. And after they see these numbers, everybody kind of nodding their head. Well, seems reasonable. And then relative cost actually plays a bigger role here. But in this case, it's relatively easy to forecast. Again, we used a very good cost analyst, and she was able to come up with very solid projection. So with this setup, what does the bar look like? First of all, the bar is going to be based on the weighted OS effect estimate. So we give observed OS a weight of W, and PFS, the OS effect derived from PFS effect weight of 1.5, 1 minus W. But that has to be multiplied by 0.5 because PFS effect is usually stronger than OS effect. So this effect over here means, roughly speaking, well, mathematically speaking, it's log hazard ratio. In approximately, you know, it's just a hazard reduction. And based on the projected number of events for both OS and PFS, we arrive at a weight of 0.15. Basically, we give OS a 15% weight and the PFS 85% weight. And it turns out that the optimal bar for the hazard ratio is about 0.91 at end of phase 2. And if we really want to go faster, we would need a hazard ratio about 0.84. Again, this all refers to the estimate OS effect by this weighted estimate. And this weighted estimate is only valid if the assumption about OS effect and the PFS effect is valid. So before you make decision, we have to also check the assumption. And so this bar here basically is the historical relationship between PFS and OS effect. And then the bar here, the curved bar line basically is just the corresponds to the hazard ratio 0.91 for the weighted estimate. And of course, x-axis is just observed hazard ratio of PFS, and y-axis is observed hazard ratio for OS. And in order to make a go no-go decision, the data has to fall under this lower bottom quadrant. So what this means is that if you have a strong PFS, like if you have a hazard ratio of 0.6, as long as the OS is positive, it doesn't you have a go decision. Although mathematically a negative OS effect still gives you a positive OS projection. It is some from purely statistical point of view, but given that that kind of is a violation of the relationship, and besides, it doesn't make much clinical sense, right? So you do need to be positive. But on the other hand, if your PFS is moderate, like here, like 0.8, 0.9, but as long in that case, your OS effect on the other hand is very strong, like 0.6, 0.5, despite small sample size, you may still go at risk, of course, just like for any phase 3 study. And because this design has explicitly and incorporate all the major consideration of a trial design into simple equation, and results were reasonable and very convincing to all the parties involved. When it was presented by Linda to a big audience in Boston at a premier oncology conference, it was ranked one of the top 3 best presentations out of 80. And during this period of time, I also got some fame. I was invited to chair a workshop specifically dedicated to the cost-benefit ratio analysis at the annual meeting of American Anti-Cancer Research in San Diego. Now, let's apply this technique to some other decision-making scenarios. So the first question we want to ask ourselves is how to detect signals after phase 1 dose finding. In oncology, once a drug has passed those finding, it can potentially be tested in multiple indications. And for a large company like Merck, we also have a lot of interest in our drugs. And of course, each drug can be also tested at different lines of therapy and can be combined with different drugs for each tumor indication. And further, the drug can be also further tested in biomarker positive or biomarker negative, or all common patients. It can further kind of complicates. But the bottom line is that there is a budget constraint. How to basically invest more cost effectively, given those uncertainties as to which combination or which tumor, which line of therapy is the best for this drug. So we statisticians, live and breathe with type 1, type 2 errors. And today, let me talk a little bit about type 3 error missed opportunities. Just to start with a very simple example. Let's say you with the same total sample size, one may conduct a phase 2 trial with 85% power at 5% alpha, or 2 trials with 60% power at 5% alpha each. If we do a quick calculation, you may notice that it has almost identical sample size. The question is, well, which strategy is more efficient given all the unknowns? If you look at the two-trial approach, the power of either success, despite lower power, is still at about 84%, which is comparable to 85%. However, if we look at the number of successes weighted by, of course, the power, then the two-trial approach is superior to one-trial approach because if both drugs are active, you may be able to get 1.2 out of them, although it's ridiculous to say 1.2 out of 2, but just that, on average, you do have a efficiency gain in terms of number of expected tumor indications when you compare to the one-trial approach. And the improvement is appreciable if we divide 1.2 by 0.85. So again, that means that in light of uncertainty, it's actually better strategy from kind of portfolio management point of view to conduct two. And it can be further shown that it's not like the more the better. There is certain kind of mathematical, if it's too much, then it's actually, it can backfire. Now let's just go a little deeper into this example. Let's further assume that you have two tumor indications of similar interest. Now in this case, I just revert back to the traditional setup of 5%, 20%. The consensus is that the one indication is more likely to be active with 30% with 13% probability and versus the other that has only 20% probability. The respective two respective phase 3 trials will have the same design, same cost, just for simplicity and for fun. Now let's consider two scenarios. In the first scenario, both indications have the same value. There's no difference. But in the second scenario, the one with the lower probability of success has a 50% higher value. So in this case, what does the optimal strategy look like? How to basically allocate resources appropriately across these two indications. So that's the outcome. So when the two indications have equal value that corresponds to the left side panel, whether you conduct one trial or two depends on the relative cost of phase 2 to phase 3. If the phase 3 cost is expensive, meaning that the C2 versus C3 ratio is small, you choose one trial. And y-axis is optimal resource allocation. You choose your 100% resource is allocated to one trial. Basically, you choose just one study. And this study, not surprisingly, has the highest chance for success. You choose the one that has better chance, given that your phase 3 is going to be very expensive. Just give your best one the best shot. But if the relative cost starts to relative cost of phase 3 starts to get smaller, which means C2 C3 ratio gets bigger, you can start to entertain the idea of splitting the resource with a little more allocated to the one with better POS than the one with lower POS. What about the lower one has a higher value? Then basically the curve, the shape of the curve has flipped. Now let's answer another question that seems to be statistically unaddressable. Basically, how many tumor cohorts should be studied after dose finding during the first in human efficacy expansion? So in oncology, what we normally do is you have you have a first wave efficacy screening in a set of tumor types of those findings. If none of them is positive, you terminate the program. If anyone is positive, then you go to next step. First of all, you follow up with either phase 2 or adaptive phase 2/3 or phase 3 in those with a positive outcome. The positivity gives you a sense that the drug is working. So now it opens the door for you to explore a second wave of tumor types. So from high level point of view, that's what we do normally in the oncology field. So when I posed this question to a newcomer who joined Merck very recently, her immediate response was, well, it's probably in the 3 to 5. And very interestingly, if we go through a very intensive statistical analysis, as I just did before, of course, in this case, it becomes a little more complicated. You indeed arrive at optimal strategy of 3 to 5. So this is discussion of my presentation. So first of all, I want to emphasize that while prior data on drug activity is crucial for deciding the aggressiveness of a phase 2/3 program, whether it's phase 2, adaptive phase 2/3, or phase 3, once the program is launched, it becomes less important than the relative cost saving in deciding whether to continue. Because as you can tell, the go-no-go bar really is reasonably robust to the prior assumption about P. And it's often unknown whether drug has a target effect or not. But projecting the costs is easy, allowing for generating robust baseline decision rules based on the BCR analysis. And I also kind of mentioned that a lot of programs, phase 2, phase 3 programs, may benefit from the 2-in-1 strategy. And when it comes down to portfolio level management, while phase 1, 2 arrows often focus, the phase 3 type 3 arrow is a concern. And probably how to properly balancing all three is key to long-term success. So this is kind of my comment that a comment that I hope that some of the statisticians working in the oncology field can take into heart, because the IO-like aggressive approach to oncology drug development is just unsustainable. And this perspective hopefully gives you opportunity to kind of think harder and so that you can help your team to make more objective and rational decision. You don't have to literally apply benefit cost ratio analysis, just that the drug development, as you can tell, is not just about analysis reporting. It does the high level statistical strategy can potentially make a difference. And what I'm providing, of course, is just for baseline decision purpose. And to that, I have to caveat that oftentimes go-no-go decisions not only depends on strength of signal, but also it depends on commercial value, strategic fit, etc. And similarly, whenever it comes down to whether to continue a program, whether it's phase 2 to phase 3 transition or futility analysis, it also has to depend on regulatory, clinical, and other statistical consideration. Of course, operational consideration is also part of that. One thing we try to avoid is to include those prior investment expenses into the kind of calculus of the go-no-go decision for the current situation. If you carry those things to the current position, you are basically committing the so-called sunk cost fallacy. Basically, past is past, shouldn't linger on that. By the same token, preclinical drug activity, no matter how strong it is, by the time you are making this kind of go-no-go, continuing a program or not, this sort of decision, it should entirely based on what you have observed, based on this very program, very study, instead of this historical kind of background. And last but not least, I want to emphasize that whenever you are trying to make any go-no-go decision or any decision of continuation or not mid-trial, the relationship or treatment effect between the early and the point you are primarily based upon for decision making and the clinical endpoint of the phase 3 study plays a key role. And we often have to be modest about its uncertainty. So hopefully I didn't overburden you with any mathematical details. If you are indeed interested, you can take a look at the references provided here. But the key method is quite easy to understand. Anyway, with that, I can entertain any questions. We still have 7 minutes left.
Thanks, Kang, for very insightful presentation. So let's start the Q&A session. Anyone wants to ask a question, please unmute your line.
So, Cong, this is Bob Beckman. Hi.
Hi, Bob.
Bob.
I just want to say two things. First of all, I think of all the things that we did together, to me, the thing that I want to just reemphasize is the type 3 error, which is also often not considered. And we found that that in some cases, if the number of reasonable phase 2 studies exceeded the budget, such that studies were getting cut, that it was better to do smaller phase 2 studies, but not to lower the bar and be aggressive, but to actually lower the power of those phase 2 studies, as you illustrated with the 60% power studies. I think that is still a very counterintuitive thing that I just wanted to emphasize. The other thing I wanted to mention is that you talked about the optimal number of drugs to screen in a 2-stage screening process. And I just wanted to point out, I know you know this, that Linshin He from Novartis and I investigated that same problem, and in some cases made strikingly different assumptions in our model than yours, but basically came out with the same answer. I think we said the optimum was 2 to 4, and you said 3 to 5. So that's probably within the validity of the assumptions, and it's probably 3 to 4. But that seems like a very robust conclusion, which is just another thing I wanted to point out.
Thank you, Bob. Any other questions?
Yeah, hi. My name is Carl Domachowski, Astellas. I'm looking at, you say that IP activity, preclinical IP activity should play no role in your go-no-go decision criteria in phase 2, 3. Is that specifically for IO drugs, or is that hold for small molecules?
Well, what I'm trying to kind of preach here is that past is past, and whenever you are trying to make a decision, you should based on present day data, like from again high level point of view. So if you are making a go no go decision from phase 1 to phase 2, 3, that's for sake argument, you should focus on phase 1. You shouldn't even care about what you have observed in preclinical setting. And for this matter, how much money you have spent in generating the preclinical data is irrelevant. I sometimes, the reason why I emphasize this is that a lot of people feel like, I already spent so much money in this program. For example, we acquired this drug from small biotech company after spending $1 billion. Now you tell me that since the phase 1 data doesn't look so well, I should abandon it. They cannot swallow that pill. And for similarly, they already started phase 3 based upon very limited data from that small company they acquired. And the phase 3 at the time of futility analysis, the data actually shows very bad. It doesn't have the promise they hoped for. But now, since they already spent so much money, they feel like we should continue. This kind of this is basically what I mean by sunk cost fallacy, and it's very rational. And since you brought that up, let me just kind of elaborate a little bit. Bob and I, we can proudly claim that we were the first to publish this method. But then a few years later, actually, there are two researchers, if I remember correctly. So fortunately, I have Bob here. He has a better memory. Two researchers from Eli Lilly. They published essentially the same method on Nature Review Drug Discovery. Except that in the denominator they added the various other cost, like basically it's some cost to the equation. And I think Bob, I remember that you wrote a complaint to the wrote a letter to complain about it to the journal, but the journal didn't respond. But that's not important. But more important is that this is apparently inappropriate to incorporate, because the past is past. You probably have made a wrong decision in the first place. Same can be said about drug activity, like in preclinical setting. It doesn't matter whether how many mice the drug has killed, has cured or something. It doesn't matter.
I don't remember actually writing to Nature Reviews Drug Discovery, and in fact, we had published our concept of the type 3 error in an earlier article in Nature Reviews Drug Discovery. So there were two articles about the same topic there. But what I would add to this, Cong, and for the audience, I'm a physician with a lifelong interest in math, and when I came to Cong with that very simple equation, he did much more with it than I could have done mathematically. But if I put my clinician hat on for the moment, when I was in industry, I was responsible for evaluating the promise of drugs and whether they should be invested in human beings based on preclinical data. And I'm sorry to say that my experience is that it's not completely worthless, but it is somewhat overrated in its predictive power. And time and time again, especially in oncology, where most drugs fail, drugs that appeared to have airtight cases in the preclinic failed in the clinic. And so I just think of course the sunk cost thing is a complete fallacy, but even in terms of this past versus future thing, I would trust clinical observations from the past more than I would trust our preclinical models.
Thanks, Bob, for the additional comment. Any other questions?
Okay, we are at 1 minute past the hour. If no more questions, we can end the call. Thanks, Cong again, for very nice presentation, and thanks everyone for joining our webinar today. Stay tuned for our webinar in March.
Thank you.
Bye.
Bye.
Thank you.
Bye.
Thank you.